Let me start by thanking Brian and the other symposium organizers for allowing me to speak in this session on understudied G-protein coupled receptors, especially in light of the fact that I will be talking about a very well-studied GPCR, the angiotensin II type 1 receptor, or AT1R. But I hope you will hear me out in spite of that, because many of the themes we see in how this receptor works apply to all of the scores of understudied GPCRs out there, and therefore I think it's really important for this audience to appreciate them. The key idea I hope you take away today is that GPCRs are conformationally heterogeneous proteins that can sample a number of different states, and because of this, different ligands targeting the same GPCR can do more than just turn the receptor on or off. They can show much more nuanced pharmacologies. The reason for this is largely because GPCRs interact with multiple types of proteins. Of course, what we most commonly think of is the fact that after agonist activation, they activate heterotrimeric G protein signaling, as shown here. But in addition, after agonist binding, GPCRs are phosphorylated by GRKs, that's GPCR kinases, which promote the recruitment of beta-arrestins. Beta-arrestins have a number of different functions. First of all, they can block further G protein activation, what we call desensitization. They also act as adapters for endocytic machinery, promoting receptor internalization, and they can scaffold kinases, allowing them to activate signaling cascades that are distinct and separable from those turned on by G proteins. Now, these different signaling pathways have different cellular and physiological consequences. And I'll just use the AT1R as an example since this is our model system today. When it gets turned on by its endogenous agonist, angiotensin II or Ang2, this leads to GQ signaling that ultimately results in vasoconstriction. That's why antagonists of this receptor, angiotensin receptor blockers or ARBs, which shut down all signaling through the receptor, are used clinically for the treatment of hypertension. But Ang2 also activates beta-arrestin-mediated pathways that have positive cardiac effects. So it was very exciting when it was discovered that certain ligands of this receptor can activate beta-arrestin pathways, but not GQ pathways. And this is what we would call a beta-arrestin biased ligand. And such ligands for the AT1R are currently being tested for acute respiratory distress syndrome associated with severe cases of COVID-19 because of the anti-inflammatory and positive cardiopulmonary effects that they have through beta-arrestin pathways. And of course, I think everyone appreciates that by now, many ligands biased either towards beta-arrestin or G protein signaling have been discovered for many other GPCRs. In many cases, it does not take huge changes in the structure of a ligand for it to show these biased signaling properties. So in the case of the AT1R biased ligands, they are all close analogs of angiotensin II itself, which is an octapeptide with the sequence shown here. The beta-arrestin biased ligands, several of which are shown, what's common about all of them is that they have changes in that 8th final residue. Instead of this big bulky aromatic phenylalanine, they have something small like an alanine residue or even an outright deletion of that last residue. There are also GQ biased ligands known for this receptor. Now these ligands retain the ability to couple to beta-arrestin like Ang2, but they show an enhanced coupling to GQ. These GQ biased ligands, they retain that last phenylalanine, but they have changes closer to the N-terminus of this peptide, around arginine 2. So it seems pretty intuitive that biased agonists must change the conformations of GPCRs in a way that's somehow different from other agonists. But despite the explosion of structural information on GPCRs in the past decade, it's been difficult to reconstruct a detailed picture of exactly what these ligands are doing from start to finish. So GPCRs, of course, have 7 membrane-spanning helices that form a bundle. And what happens as you move from the inactive conformation in gray to the active conformation in orange is that agonist binding in the extracellular ligand binding pocket causes small but significant changes in its architecture. This is propagated through the rearrangement of residues in the receptor core and ultimately results in much larger scale conformations in the intracellular transducer binding region. If we look up at that region now, as though we were underneath the receptor, the biggest change, what's really considered to be the hallmark of GPCR activation, is this outward movement of TM6, typically by about 10 angstroms. That's accompanied by smaller movements of TM5 and TM7. The net result is a configuration of the site that's far more open and accessible so that G proteins and beta arrestins can get in. But even with all the scores of structures of GPCRs, both in their inactive and active states saw so far, these changes have been pretty well conserved. So again, this begs the question, what is it that biased agonists do that's different? So some of the best information previously came from the Roth group. They solved structures of a serotonin receptor, 5-HT2B, in complex with a series of ligands, including antagonists and agonists biased towards either arrestin or GQ signaling. And in doing so, this allowed them to hone in on specific changes that each type of ligand induced in the extracellular ligand binding pocket. But what's been more difficult for the field as a whole is figuring out how these differences in the ligand binding pocket can be propagated all the way through the receptor, from through these middle core regions and all the way down to the transducer binding pocket. So to try to understand this for the angiotensin receptor, we and our collaborators used a couple of different biophysical techniques to provide information on different regions of the receptor. To get a high-resolution view of the ligand binding site and the receptor core, I'll be telling you about how we used X-ray crystallography, solving structures of the AT1R in complex with a series of different ligands. Then we used double electron resonance spectroscopy to look at the intracellular regions. This is a method that allows us to see the full conformational heterogeneity of everything that's going on there. Since only structures of the AT1R bound to antagonists had been previously solved by other groups, we and our collaborators at the Cruz Lab at Harvard obtained structures of the receptor with angiotensin 2, with a weak partial agonist called S1I8, and with two fully arrestin-biased agonists. While I don't have time to go into it today, this was made possible by using an antibody fragment or nanobody obtained by an in vitro yeast display selection to stabilize the intracellular conformation of the receptor in an activated conformation. So as we focus in on different regions of the receptor, I'll put this icon in the lower corner to remind you approximately what area we're looking at and sort of the viewing angle. So all of our 4 peptide ligands induced similar changes in the architecture of the ligand binding site, as shown here. So the receptor is shown as a surface representation in these two figures, and we're outlining the sides of the ligand binding pocket in the yellow dashes. And so if we model where one of our peptide ligands would be onto the inactive structures that are actually bound to antagonists, you can see how much space is left around it. In all of our peptide-bound structures, the receptor is really clamped down around the peptide, and the N-terminus is up at the top, C-terminus is down at the bottom. Now, again, this happens in all 4 of our structures. And in fact, if we overlay even our most extreme ones, Ang2 and our arrestin-biased ligands, you can see, looking at both the side and from this top view, you can see that there's really not much of a difference. Of course, what we want to see is something different between our biased ligands and Ang2, which can actually strongly activate GQ. Turns out there was only one difference, and that's right at the bottom of the ligand binding pocket. So at the base of the pocket, there are two residues, leucine 112 and tyrosine 292. And you can see if we compare the inactive in gray and one of our biased structures in blue, you can see that there's not much change in those two positions. This is the C terminus of the ligand up above, and you can see that it's fairly well removed. But if you remember, what's different about Ang2 is that it ends in this big bulky phenylalanine. This clashes with where that leucine residue was before, causing it only in the Ang2 structure to rotate over, hit where the tyrosine 292 was before, and cause that to become highly dynamic so that we can't even see it. Now, I'm going to show you another view of that same event, looking at it from the top now, to show you that as that leucine rotates, the entire TM3 that it's on is also rotating. And so in other words, the adjacent residue, asparagine 111, is also rotating with it. So why does this matter? Well, this is how the difference is getting propagated away from the ligand binding site and down into the receptor core. So if we, in the inactive state of the receptor core is stabilized by a network of polar interactions that between TM7 and TM3, TM7 and TM2. So between TMs that are on the opposite side of the receptor. In the fluorescent-biased ligand structures, TM7 has moved. That's the result of the changes in the overall architecture of the ligand binding site that we were looking before, how it clamps down on the ligands. Because of this movement of TM7, that moves these two asparagines away from the network so they can no longer form these stabilizing interactions, sort of severing those connections across the receptor. Then, asparagine 111 is also initially part of that network. So after this, this is the residue that we said on TM3 rotates. As a result, it's now facing in the Ang2 structure only. It's now completely removed from this network, kind of severing those last inter-TM connections among it. So, what we've seen so far is that both in the ligand binding site and in the receptor core, with the fluorescent-biased ligands, we see sort of a partially activated conformation. And then with Ang2, we see a more fully activated conformation. So, change in the architecture with the fluorescent-biased ligands, this extra switch flipping sort of with Ang2, partially severed network in the receptor core with the biased ligands, more fully severed with angiotensin 2. So what happens next? Of course, what we want to see is how this affects the intracellular regions where G protein and beta-arrestins actually bind. But this is where we get stuck with our structures, because if you remember, we had to put this nanobody there to stabilize everything in a single conformation. So to try and get into this question, we collaborated with the Hubble group at UCLA and the Kabilka group at Stanford to use double electron-electron resonance spectroscopy to look at the intracellular conformations. So this is an electron paramagnetic resonance technique, meaning that it's sensitive to unpaired electrons. So if we put nitroxides that have unpaired electrons, if we put two of these labels on different sites of our protein, we can measure the distance between them. So for example, if we want to look at this outward movement of TM6 that we say it says the hallmark of GPCR activation, we can see that in the absence of ligand for the AT1R, we've got two major populations at about 30 to 35 angstroms. But then when we put angiotensin 2 on the receptor, about 2/3 of the population moves to this much longer distance, reflecting this outward movement. So we did this not only for TM6, but for 10 different pairs of residues throughout the intracellular regions of the receptor, and ran these with each of these with 10 different ligands. So when we put all these data together, we can get several types of information. So first of all, we could see that there was not simply two conformations, an inactive and activated, but 4 distinct conformations. Second, we could determine the fraction of the receptor population in each conformation under each ligand condition. So what are these different conformations and where do they show up? So the first conformation we saw we call the closed conformation, and it's very similar to what we see in inactive crystal structures. And so the position of our different labels are shown in the colored spheres here, and you can see that they are the TM6. If you compare this to an inactive structure that's shown in the ribbons, TM6 is still in this inward position, and overall the transducer binding site seems to be fairly well closed. Again, very similar to inactive crystal structures. We see where this shows up with different ligands. It's dominant in the apo state, that's the absence of any ligands, and it's further enriched by the addition of antagonists. Makes sense. The second conformation we saw we called the open conformation because it's very similar to the canonical active GPCR conformation. So TM6 has now moved out. We see other typical rearrangements like this apparent rotation of TM5 and this inward motion of TM7. So this conformation is very minimal in the absence of ligand or antagonist, and even with the Barr-biased ligands. But where you start seeing it increase is with Ang2, and even more strongly with these GQ-biased agonists. So it seems like the more coupling you have to GQ, the stronger this population is. So where does that leave the Barr-biased agonists? Well, they're stabilizing two other conformations that we call occluded because they don't seem to have all the characteristics of the canonical active conformation. So in the first of these, what we call the occluded 1 conformation, TM6 is actually still in a fairly inward position if you follow the arrows. But other conformational changes have occurred, such as this inward movement of TM7, and helix 8 as well. The second one is actually TM6 has already undergone this large outward movement, but there's other differences from the active conformation. So for example, TM5 hasn't rotated as much, and ICL2 seems to have undergone this conformational change. So it seems like it's not quite as accessible as the canonical active conformation. So again, these two are stabilized in various mixtures by all these different beta-arrestin biased ligands. And interestingly, even angiotensin II seems to stabilize a considerable amount of it. So if we put this together now, what seems to be happening is that biased ligands aren't stabilizing conformations that are unique or different than the endogenous agonists per se. The reason we can observe this bias seems to be that Ang2 itself stabilizes a mixture of both fully activated and partially activated conformations, as shown here. So when you have a ligand that shifts the equilibrium away from the fully activated conformations to some of these more partially activated conformations, that seems to result in Barr-biased bias. On the other hand, if you shift more towards these fully active conformations, that seems to result in GQ bias. And if we step back and look at everything we've seen so far, it's really been a consistent theme in how these biased ligands operate at every level of the receptor. In the ligand-binding site, Barr-biased ligands stabilize only this partially activated conformation, Ang2 flip this extra switch. In the receptor core, Barr-biased ligands partially destabilize this network, Ang2 did so more fully, and again, something similar in the intracellular binding pocket. And we think what this is telling us is that GQ is very highly specific for this fully active, fully open conformation, while Barr-biased is much more promiscuous, able to recognize a larger range of conformations. So, well, you're probably wondering, does this matter for your understudied GPCR? Well, GPCRs are much less structurally conserved in the ligand-binding site, where they bind to many different types of ligands, but far more conserved in the intracellular regions. So we expect that we'll see that different receptors have different types of switches that ligands activate, but these will kind of have different paths to the same goal and stabilize very similar intracellular conformations. So remember that as you dissect the signaling of your favorite GPCR, remember that biased ligands may allow you to pharmacologically dissect them, and hopefully we'll be able to help you come up with ways to do so rationally in the future based on mechanism. I'd like to thank my postdoctoral advisor, Dean Stouse in the laboratory, our collaborators in the Cruz, Hubble, and Kabilka labs especially, and thank you.